Warm greetings to all of you. I welcome you all on the National Science Day 2024 Applications of Biotechnology for Viksit Bharat. The title for the talk today is Therapeutics for Protein Misfolding Diseases. In order to be functionally active, a protein has to acquire a unique three-dimensional conformation via a complicated folding pathway, which is described by the primary amino acid sequence and the local cellular environment. Protein folding is vital for a living organism because it adds flesh to the gene skeleton. A small error in the folding process results in a misfolded structure which can sometimes be lethal. However, within the cellular environment, which is highly viscous, many proteins cannot fold properly by themselves and require the assistance of a special kind of ubiquitous protein, the molecular chaperones. Molecular chaperones assist other proteins to achieve a functionally active three-dimensional structure and thus prevent the formation of a misfolded or aggregated structure, essentially enhancing folding efficiency by influencing the kinetics of the process and inhibiting events that lead to unproductive endpoints, example aggregation. Chaperones are located at various points in the cell and interact with nascent polypeptides during synthesis and translocation to different cellular compartments. Chaperones are able to distinguish between the native and non-native states of target proteins, but how they discriminate between correctly and incorrectly folded proteins and how they selectively retain and target the latter for degradation is yet to be understood. Proteins that are not able to achieve the native state due either to an unwanted mutation in their amino acid sequence or simply because of an error in the folding process are recognized as misfolded and subsequently targeted to a degradation pathway. This is referred to as protein quality control system and is composed of two components, molecular chaperones and the ubiquitin proteasome system. The QC system plays a critical role in cell function and survival. A special class of chaperone, for example, calnexin, forms a part of the quality control monitors that recognize and target abnormally folded proteins for rapid degradation. In almost all protein misfolding disorders, an error in folding occurs because of either an undesirable mutation in the polypeptide or, in few cases, some less known reason. The harmful effect of the misfolded protein may be due to loss of function as observed in cystic fibrosis and alpha-1 antitrypsin deficiency, or deleterious gain of function as seen in many neurodegenerative diseases such as Alzheimer's disease, Parkinson's disease, and Huntington's disease, in which protein misfolding results in formation of harmful amyloid. Protein aggregates are sometimes converted to a fibrillar structure containing a large number of intermolecular hydrogen bonds, which is highly insoluble. These are commonly called amyloids, and their accumulation occasionally results in a plaque-like structure. In some cases, the mutations are so severe that they render the gene product biologically inactive. In other cases, however, the mutations are relatively minor and the resulting proteins show only a partial loss of normal activity. Despite having partial biological activity, these mutant proteins are not delivered to the correct location, either inside the cell or in the extracellular space. One example of disease involving abnormal protein trafficking is alpha-1 antitrypsin deficiency. In almost all cases of protein misfolding mediated disorders, mutation in the gene is very common. However, more frequent amyloid-related neurodegenerative diseases are characterized by the appearance of a toxic function caused by the misfolded protein. One or more of these chaperones' activities result in the prevention or suppression of a few devastating neurodegenerative diseases. Reduction in the intracellular level of chaperones results in an increase in abnormally folded proteins inside the cell. Therefore, toxicity in different neurodegenerative disorders may result from an imbalance between normal chaperone capacity and the production of misfolded protein species. Increased chaperone expression can suppress the neurotoxicity caused by protein misfolding, suggesting that chaperones could be used as possible therapeutic agents. Natural, chemical chaperones have been shown to be the promising agents for the control of many protein conformation disorders. These diseases include cystic fibrosis, Alzheimer's disease, Parkinson's disease, Huntington's disease, etc. Protein misfolding and its pathogenic consequences have become an important issue over the last 2 decades. According to the prion researcher Suzanne Lindquist, protein misfolding could be involved in up to half of the all human diseases. Protein misfolding is also responsible for many of the p53 mediated cancers, which are also the result of incorrect protein folding. Many cancers and other protein misfolding disorders caused by mutations in proteins are present in the table that are key regulators of growth and differentiation. Structural changes in few proteins subsequently lead to aggregated masses, which occasionally result in neurotoxicity and cell death. Hooper reported that aggregated misfolded proteins become neurotoxic because of an inhibition of proteasome function. Sarmi suggested a chaperone overload hypothesis, which explains that with aging there is an overburden of accumulated misfolding protein that prevents molecular chaperones from repairing phenotypically silent mutations which might cause disease. It has been shown that the yield of correctly folded protein obtained from in vitro refolding is low due to the formation of thermodynamically stable folding intermediates. These conformations are called dead-end conformations and are off-pathway intermediates. They generally lead to the formation of insoluble aggregates that may eventually cause different degenerative diseases. Classic examples of these degenerative diseases is cystic fibrosis. A common feature of almost all protein conformational diseases is the formation of an aggregate caused by destabilization of the alpha helical structure and the simultaneous formation of a beta sheet. These beta sheets are formed between alternating peptide strands. Linkages between these strands result from hydrogen bonding between the aligned pleated structures. Such beta linkages with the pleated strand from one molecule being inserted into a pleated sheet of the next lead to hydrogen bond formation between the molecules. The prerequisites for beta linkage formation are the presence of donor peptide sequence that can adopt a pleated structure and a beta sheet that can act as an acceptor for extra strands. Protein aggregation is an inevitable consequence of acellular existence, and these aggregates are oligomeric complexes of a non-native conformers that arise from intermolecular interactions among structured and kinetically trapped intermediates in the protein folding or assembly pathway. Protein aggregation is facilitated by partial unfolding during thermal and oxidative stress and by alterations in the primary structure caused by mutation, RNA modification, or translational misincorporation. Protein aggregates can be either structured, that is amyloid, or they can be amorphous. In either case, they are insoluble and metabolically stable in the physiological environment. For various diseases associated with protein misfolding, one or more proteins are converted from the native structure to an aggregated mass, which is commonly called an amyloid. The net accumulation of toxic protein aggregates in the cell depends on the stability, compactness, and hydrophobic exposure of the aggregates, as well as on the rate of protein synthesis in the cell. The accumulation of toxic aggregates in the cell depends on chaperone expression and protease networks. Environmental stress may induce the synthesis of higher levels of chaperones and proteases in the cell, which can be better removed toxic aggregates. Fibrillar amyloids are commonly extracellular, but intracellular fibrillar deposits are also seen in patients. Example, intracellular bundles of neurofibrillary tangles in AD. Although the initial process might be different in different diseases, a common trend is that during the formation of aggregates, alpha helical domains disappear, leading to an increase of beta sheet dominated secondary structure. Recently, many other physiological disorders have been recognized as being caused by the formation of protein aggregation, which subsequently forms a plaque-like structure containing a large number of amyloid fibrils. These are polymerized to cross beta sheet structures with the beta strands arranged perpendicular to the long axis of the fiber. Living organisms, that is, many plants, animals, and microorganisms, have evolved mechanisms to adapt themselves against harsh environmental conditions that normally denature macromolecules. Accumulation of small organic molecules called osmolytes is one such strategy adapted by living organisms to combat denaturing stress conditions. These stabilizing osmolytes, also known as chemical chaperones, are grouped into three major classes: polyols, amino acids and their derivatives, and methylammonium compounds. Osmolytes can also be classified as compatible osmolytes and counteracting osmolytes depending upon their ability to affect protein function and stability. The property that differentiates compatible osmolytes from counteracting osmolytes is what effect these two classes of osmolytes have on stability and function of protein. Compatible osmolytes increase stability of protein under denaturing conditions but does not affect the function of protein. Counteracting osmolytes, on the other hand, confer protection against the inactivating and destabilizing effects of urea. Some organs and organisms use osmolytes that have ability to counteract the destabilizing and denaturing effects that urea has on their proteins. Osmolytes are termed protective since they have the ability to confer stabilization on proteins under denaturing conditions without significantly affecting their function. Osmolytes have also been implicated in stabilizing proteins that are less stable and also cause refolding of proteins that are malfolded. Applications and implications of osmolytes in various diseases have also been extensively investigated. The native three-dimensional protein conformations play a crucial role in viability of cell under physiological conditions. Different conformations are adopted by proteins as dictated by the functional and cellular requirements of the cells. For instance, proteins that tend to degrade generally acquire an unstable conformation and are marked by ubiquitin, while proteins not intended for degradation are not marked by ubiquitin. This equilibrium is very crucial for cells to maintain the homeostasis of normal proteins. Even a minute imbalance may lead to proteopathy. Osmolytes, by their ability to stabilize or destabilize proteins, modulate protein homeostasis. Cell viability is determined by the very delicate and correct balance that exists between degradation and proper folding of proteins that are either unstable or malfolded. Proper assembly and correct folding of newly synthesized protein in early events of the secretory pathway, the nucleoplasm and the cytosol dictate its quality. Stabilizing osmolytes are believed to help in surveillance of the misfolded and unstable macromolecules, guide them to attain proper folded conformation and structure, and therefore increase unstable protein homeostasis. In contrast, destabilizing osmolytes act like denaturants, thereby destabilizing proteins and ultimately helping in promoting degradation of proteins that are aggregated or are excessively stable. Involvement of osmolytes in order to keep a regulatory check on homeostasis of proteins, therefore, is logical. The importance of osmolytes in regulating protein homeostasis has well been demonstrated by diverse number of studies. The function of many misfolded proteins can be rescued by refolding or protecting degradation attack by proteases in presence of osmolytes. If the property of accumulating osmolytes could be changed inside the intracellular environment, this can simply lead to changes in protein stability. For instance, betaine, when present at low concentrations, acts as a refolding inducer, whereas it turns to be a denaturant at higher concentrations. The disorders and diseases related to age or those that have genetic background are in fact issues of protein turnover or homeostasis, which may be due to enhanced proteasomal degradation or growth of high-order aggregates. The term protein conformational diseases has been aptly used to group all these diseases. This encompasses diseases like Huntington, serpin deficient disorders, etc. Treatment of these conformationally less stable proteins by suitable proteostatic modulators, the osmolytes, may be a unique strategy for the therapeutic intervention of diseases due to proteopathic disorders. The denaturant-like properties of destabilizing osmolytes can be potentially used to remove toxic protein aggregates formed by mutant proteins. In conformity with this notion, osmolyte like lysine or arginine and the small peptides are conventionally implicated to help to increase the solubility of inclusion bodies in bacteria and protein aggregates. In contrast, the stabilizing osmolytes serve as outstanding protein folders. Thus, the role of osmolytes in correcting mutant protein function is crucial. Furthermore, many osmolytes bind to specific proteins and help to modulate stability and hence function as a strategy for post-translational modification. For instance, there are existence of many proteins that bind arginine and regulate function. The methyl donor betaine has also been reported to bind to many proteins and help to regulate function of many proteins. The misfolded proteins expose hydrophobic segments that are normally buried in the core of their native conformation, and this positively leads to the formation of intermolecular binding and subsequent aggregation. The oligomers and aggregates are formed when the proteins that have misfolded in the cells reach a specific critical concentration. The partial unfolding of proteins during oxidative stress or thermal stress and by changes induced in the primary structure that are a result of mutations and RNA modification or wrong incorporation of amino acids during translation all facilitate aggregation of proteins. The aggregates of protein are insoluble and metabolically stable and can assume either a structured conformation, that is amyloid, or an amorphous conformation in physiological environment. A lot of effort has been put so far to get an essential insight of the basic origin and factors that affect the process of protein aggregation, leading to development of human diseases. Indeed, aggregation kinetics is sigmoid and consists of three important steps: lag phase, that corresponds to conformational transformation in the native monomer resulting in generation of aggregation prone intermediates; the exponential phase, that corresponds to self association or oligomerization of the monomers; and the stationary phase, that leads to appearance of mature aggregates or fibrils. The intracellular deposition of proteinaceous toxic aggregates corresponds to the rate of synthesis of protein and also on certain parameters: thermodynamic stability, structural compactness, and exposition of apolar moiety of the aggregated proteins. A common feature observed during the formation of aggregates in protein aggregation diseases is the disappearance of alpha helical structures, resulting in enhanced beta sheet conformation. The formation of protein aggregates has occupied center stage in medical field since in vivo deposition of aggregates has been attributed to the pathogenesis of various diseases, for instance neurodegenerative, metabolic, cardiovascular diseases, etc. Different sequences and tertiary structures have been observed for proteins involved in these protein aggregation diseases. Interestingly, many peptides and proteins which are not direct participant of disease pathogenesis also form aggregates when subjected to specific conditions. Therefore, it would not be incorrect to say that it is a general behavior of all polypeptides and proteins to form aggregates. Prolin at concentrations above 3 molar was found to prevent aggregation of bovine carbonic anhydrase. Another study proposed the prolin to be aggregation protective since it was able to inhibit the aberrant interactions among polypeptide chains of protein very early into pathogenic aggregation pathways. The polyglutamine-rich polyQ type of Huntington protein deposition occurs inside the nucleus, which is the characteristic feature in the brain of patients of Huntington disease. In a study, 21-day-old mice were orally fed with 2% of trehalose solution until the day the mice were killed. The study clearly demonstrated decrease in aggregation tendency of the disease-associated polyglutamine-containing protein Huntington. This resulted in improvement in loss of motor function and also increased the lifespan of the transgenic Huntington mouse. In other studies, prolin was also found to protect lysozyme from chicken egg and creatine phosphokinase from rabbit skeletal muscle from aggregation. However, there are reports from research groups that osmolytes favor or promote protein misfolding or does not have any significant consequences on protein aggregation pathway. For instance, TMO was found to promote aggregation during refolding of RNase A. Glycine had only small effect on aggregation of fatty acid synthase, even when used at higher concentrations, and it failed to prevent aggregation of protein on lysozyme refolding. A study demonstrated that taurine was able to delay the onset of fibrillation of glucagon, a peptide hormone. It was also observed that the morphology of glucagon fibrils remained unchanged in the presence of taurine. Published results show that trehalose showed complete inhibition of fibril of A beta 40. Studies on the SUP43, which is a prion protein of yeast, in the presence of trehalose have demonstrated its aggregation inhibiting effect by maintaining the protein in partially folded state. In other studies performed on mouse model of oculopharyngeal muscular dystrophy with osmolytes, trehalose has demonstrated suppression of aggregation in such systems. The most debilitating disorders are the neurodegenerative diseases. These diseases affect thinking, movements, cognition, and memory. This diverse group of diseases is characterized by certain characteristics such as their emergence late in life, the associated extensive aberration of synapse and neuronal loss, and accumulation of aggregates or inclusions and plaques of neurons, although they have significant dissimilarities in their clinical manifestation. These deposits activate succession of key events that result in the impairment of synapses followed by death of neurons, with deleterious clinical aspects of pathogenesis of disease. However, in each pathogenic condition, the distribution and composition of protein aggregates is different. Alzheimer's disease is a disorder of brain which cannot be reversed. It has a drastic effect on person's memory and therefore hampers individuals' capability to execute normal routine. It is a well accepted fact that the major pathology of Alzheimer's is related to deposition of beta amyloid peptide in several regions of brain. The natural proteolytic cleavage of A beta protein precursor yields A beta. Three different proteases process A beta protein precursor, cleaving it at dissimilar locations and yielding different A beta peptides that ranges from 39 residues to 43 residues length. The primary building unit for insoluble A beta extracellular deposits or senile plaques in the A beta peptide, which is the neuropathological hallmark of Alzheimer's disease. Other features of Alzheimer's disease include accumulation of intracellular triangular filaments of hyperphosphorylated tau called neurofibrillary tangles. Distinct set of cytopathologies, for instance, mitochondrial dysfunction, non-functionality of cytochrome C oxidase, dimerization of A beta peptide precursor, disturbed iron metabolism, synaptic dysfunction, and oxidative insults, and energy hypometabolism revealed the cellular dysfunction in this disorder. The A beta 40, when treated with trehalose at physiological concentration, was found to inhibit aggregation behavior of A beta 40 in a concentration dependent fashion. A perfect correlation was observed with the trehalose mediated prevention of A beta aggregation and its cytotoxicity inside the neurons. From these results, it would not be incorrect to say that osmolytes can be therapeutically utilized to treat Alzheimer's disease. Osmolytes may also play a role in stabilizing the senile plaques and also protecting the release of toxic A beta oligomers. Since osmolyte levels are found to decline in Alzheimer's patient, they may also function as antioxidants. Alpha-synuclein is an intracellular, soluble, small 14 kilodalton protein. The function of this protein is unknown, and it is found abundantly in numerous places of the brain. Characteristic features of alpha-synuclein are the occurrence of stretches of acidic amino acid residues in C-terminal and the motif repetitions in the initial 93 amino acid residues. The alpha-synuclein aggregate deposits are found in the cytoplasm of glial cells and neurons and serve as pathological characteristic of many neurological disorders, including Parkinson's disease. Alpha-synuclein has been found to readily assemble into fibrils in vitro. These fibrils share similar morphological and staining characteristics as with the fibrils extracted from Parkinson's disease affected brain. Interestingly, influence of chemical chaperones, for example TMAO, on the aggregation behavior of proteins was found to depend on concentration of the chaperone used. When used in higher concentrations, it causes the alpha-synuclein to fold tightly, consequently forming oligomers that are more stable and have decreased fibrillation rate. But when TMAO was used at lower concentrations, it is found to form partially folded intermediate structures, that which accelerates fibrillation process. So in light of these experiments, the use of osmolytes for treatment of neurodegenerative disorders shall require more stringent explorations. The transmissible spongiform encephalopathies are a family of disease that comprise of spongiform encephalopathy in bovine or mad cow disease, scrapie in sheep, and various rare neuropathies in human. The hallmark of TSC pathogenesis was the assembly of an abnormally folded version of a normal cellular form of PRPC in brain cells. Griffith, for the first time, propounded a protein-only model to describe the unusual infectious behavior of TSC proteins. The term prion has been aptly used for this agent. Prions are entirely proteinaceous, consisting mainly of the PRPSC. These are experimental evidences that show that PRPC and PRPSE are two distinct conformations. However, both molecules have been derived from one primary amino acid sequence with no observable post-translational modifications. The PRPC is converted to the scrapie form through interactions between the normal PRPC and the pre-existing scrapie form of the protein, although the concrete mechanisms followed by the protein for conversion are unknown. Several workers have examined the effects of osmolyte, for example, trehalose, sucrose, glycerol, on the conversion and infectious behavior in the PRPC normal conversion process. In the absence of the scrapie form or reactions which were seeded with the scrapie form, these osmolytes did not have any effect. However, in reactions that were seeded with infectious scrapie form, DMSO demonstrated a concentration dependent influence. Trehalose and sucrose show inhibition of the pathogenic conversion of the normal prion protein. The inhibitory effects on the pathogenic conversion by osmolytes were found at excessive dose only. This result has direct physiological relevance because such osmolytic chaperones have been reported accumulate at such high levels in vivo under denaturing stress conditions. Cystathione beta synthase (CBS deficient homocystinuria) is a disorder associated with the metabolism of sulfur amino acids. It is characterized by the buildup of toxic homocysteine and its metabolites in urine and blood due to the defects in HCY metabolizing enzyme CBS. Missense mutations account for approximately 87% of all mutations in CBS. Many of these mutations are not present in the catalytic residues in CBS, but rather result in misfolded proteins which lack biological function and are destined for degradation. The clinical symptoms include dislocated optic lenses, neurodegeneration, skeletal abnormalities, and cardiovascular complications. Studies have demonstrated the basic origin of HCY toxicity is covalent adduct formation of protein on the lysine residues by homocysteine thiolactone as a process called protein N homocysteinylation. Protein N homocysteinylation has been known to result in formation of oligomers to aggregates or amyloids and ultimately loss of enzyme function. Therefore, prevention of multiple or oligomer formation and solubilization of toxic amyloid or aggregate induced by homocysteine thiolactone may help to prevent homocystinuria-induced cytotoxicity. On the other hand, strategies that can help to restore the mutational defect in CBS will also be of immense use for the treatment of homocystinuria. Only about half of the patients deficient with CBS are found to respond to vitamin B6 treatment, which causes large reduction of the toxic homocysteine levels in plasma and hence reduction of associated clinical phenotypes. It was demonstrated by Shan and Truger that the activity of I278T, which is the most predominant missense mutation found among CBS patients, can be partially restored if the C-terminal endomate binding domain is truncated or a point mutation is introduced in the C-terminal region. It was inferred from these observations that the I278T mutation affects folding, not the catalytic activity of CBS. A number of patients' fibroblast lines tested for CBS by electrophoresis in native page followed by Western blot analysis, the CBS mutants were found not to form active tetramers and accumulated as aggregates that are inactive. These misfolded CBS aggregates were also found to be devoid of heme. These data suggest that the inability of mutants to properly incorporate heme may prevent correct folding. Thus, abnormal folding is the most common pathogenic mechanisms in CBS deficiency. The role of chemical chaperones in restoring the function of mutant CBS was first demonstrated by Singh et al. They clearly demonstrated that osmolytes, if present during the early stages of folding pathway, could facilitate the proper assembly and folding of many CBS mutants in vivo as well as in vitro. They showed the chemical chaperones TMAO, glycerol, enhanced mutant I278 CBS activity and 3 other mutant CBS by facilitating formation of active tetramers through stabilization of the folded protein. These studies taken together demonstrate that mutant CBS is amenable and responsive to treatment with chemical chaperones in vivo, and this may begin a new therapeutic approach, especially for pyridoxine non-responsive CBS deficient patients. Role of osmolytes in cystic fibrosis. Cystic fibrosis is a disease condition which is a result of the unavailability of a functional form of cystic fibrosis transmembrane regulator, CFTR, at the normal cellular site. The functional role of CFTR is to act as a chloride channel in the plasma membrane, which is regulated by CMP. Thick mucus secretions in the lungs and intestines are the dominant hallmarks of this disease. The CFTR gene residues in the long arm of chromosome 7 and is nearly 250 kb of DNA. Large numbers of mutations were identified to be responsible for the functional deficiency of CFTR. The classification scheme for CFTR mutations developed demonstrates that majority of them cause the misfolding of CFTR protein. Analysis of the primary CFTR sequence has demonstrated that it is localized in the membrane and has two domains responsible for nucleotide binding, 12 transmembrane domains and a highly charged regulatory domain. In majority of patients investigated for CF, the most predominant mutation occurs when phenylalanine residue is deleted at position 508. These mutations are known as delta F508. This delta F508 mutation represents 70% of the patient's related CFTR mutant, which signifies its clinical importance. In these delta F508 mutants of CFTR, the newly synthesized delta F508 CFTR protein does not attain its native conformation and is not trafficked to the plasma membrane. Nascent CFTR mutant cannot undergo proper trafficking, gets trapped in the endoplasmic reticulum, and undergoes proteolysis by the action of molecular chaperones. The calnexin, which is a ER localized chaperone, and HSP73, which is a cytosolic chaperone, directly interact with CFTR. The portion of CFTR that is translocated to plasma membrane via the Golgi complex no longer interacts with both calnexin and HSP73. The delta F508 CFTR protein also interacts with calnexin and HSP73, but the interaction in this case is very stable. This stable interaction causes the mutant CFTR to be trapped in ER and marked for proteasomal mediated degradation. The delta F508 CFTR folding defect has been found to be temperature dependent. When cells expressing this mutant are grown at the temperature of 30 degrees centigrade or lower, the trafficking defect and functionality as a chloride conductance are well restored. This observation suggested that this deletion of phenylalanine at position 508 is very subtle and can be corrected. One strategy to correct this protein folding defect is the application of low molecular weight compounds, osmolytes. The delta F508 CFTR protein temperature-sensitive folding defect was found to be modified by chaperones such as TMAO and glycerol. It has been understood that delta F508 CFTR is functionally impaired due to inadequate maturation process and hence gets caught in ER as a non-native unstable conformation. Myoinositol, betaine, and taurine has also been found to correct the folding defect of delta F508 CFTR. Myoinositol, betaine, and taurine demonstrated reduced cytotoxicity in comparison to glycerol. When cells expressing this mutant were incubated in the presence of glycerol and other osmolytes, the level of mutant CFTR and CAMP-dependent activated chloride channel activity was found to increase. Pulse chase experiments indicated that in the presence of glycerol, there was increased maturation process, hence decreased proteasomal mediated degradation, which eventually leads to increased steady state level of the mutant protein. This was attributed to enhanced folding of the mutant CFTR. In another development, when mouse expressing this mutant was injected with TMAO subcutaneously, there was significant increase in the intestinal chloride transepithelial transport in the wild type and the mutant mice, suggesting a restoration of function of this mutant with TMAO. Such phenomenon of restoration of function to malfolded CFTR could boost the screens for novel compounds for drug discovery, which are potent in rescuing the mutant misfolding as harmless pharmacological concentrations. Role of osmolytes in diabetes mellitus. Type 2 diabetes mellitus is a metabolic disease which affects millions of people worldwide. It is characterized by chronic hyperglycemia and dyslipidemia. Type 2 diabetes mellitus has also been found to be associated with other varied complications. For instance, retinopathy, nephropathy, neuropathy, and cardiovascular abnormalities. Two hallmarks that define type 2 diabetes mellitus are insulin resistance and pancreatic beta cell failure. The pancreatic beta cells become dysfunctional and degenerate due to the formation and deposition of extracellular amyloid plaques. It was in 1901 that such amyloid deposits were described for the first time, though the component that is amyloidogenic, that is amylin, also known as islet amyloid peptide, which is abbreviated as IAPP, was extracted and sequenced many years later. IAPP is a peptide hormone which is 37 residues long and is co-produced and secreted along with another hormone, insulin. This occurs by the secretory pathway in beta cell and in 1:100 ratio, but in case of type 2 diabetes mellitus, it can increase to ratio of 1:20. During the process of translation of protein, the processing and modification of IAPP occur. The gene of IAPP is expressed as pre-pro-IAPP, which is 89 residues long. Located at the N-terminus is a long signal sequence peptide that is made of 20 amino acid residues. This signal peptide is crucial for the trafficking of the protein from the endoplasmic reticulum to the trans-Golgi network. A disulfide bridge between two cysteine residues is formed as the prepol IAPP is transported from ER to trans-Golgi network. After the pro-IAPP reaches the trans-Golgi network, cleavage of the signal peptide to yield 67 amino acid residue long pro-IPP occurs. This resultant pro-IPP is then acted upon by pro-hormone convertase enzyme for further processing. The steps involved in further processing include the cleavage of the 16 C-terminal amino acids and in the secretory vesicles and cleavage to the C-terminal basic amino acids catalyzed by carboxypeptidase E and activation of the peptidyl imidating monooxygenase complex that cleaves glycine at position 38 and results in the amidation of tyrosine at position 37. The human IAPP is a monomeric protein and is an intrinsically disordered protein and therefore consists of numerous flexible and random coil regions having a brief helix amphipathic in the N-terminal region. The region which has been found to be important for amyloid formation consists of portions 22 to 27. Literary evidence suggests that the double beta hairpin with 3 beta strands between residues 12 and 37 occurs when aggregated IAPP gets folded. However, although the primary structure of IAPP is well conserved through evolution, amyloid formation is not found to occur in all the mammalian species. In the islet of Langerhans of rodents, for instance, the pathogenesis related to deposition of IAPP amyloids has not been found. Proline mutations that act as beta sheet breaker in the most fibrogenic region have been implicated for this. Since it is co-secreted with insulin, this might have a hormonal role in regulating glucose homeostasis. The existence of a clear link between islet amyloid deposition and reduction of beta cell mass clearly signifies the pathophysiological effects of this protein. Dysfunctions and death of beta cells at different subcellular levels have been observed with increased expression level and aggregation of this protein. During upregulation of the expression level of IAPP, the unfolding protein response, which is UPR, within the ER compartment has been found to be induced by this protein. So, from these type 2 diabetes mellitus patients' autopsy reports, beta cells have been found to accumulate polyubiquitinated proteins and autophagosomes, which indicate toward the dysfunction of ubiquitin proteasome system and autophagy, the two major intracellular systems involved in protein degradation. A study investigated the fibrillation reaction of IAPP in the presence of TMAO, glycine, betaine, proline, and urea. TMAO and glycine betaine were found to induce stabilization of human IAPP protofibrils by causing retardation of fibril elongation and were inferred from THD fluorescence and ATR-FTIR spectroscopy data. However, the AFM images showing morphology of the mature fibrils in the presence of these osmolytes showed no effect of these osmolytes. The prolongation effect on the fibril elongation in the presence of TMAO was concentration dependent, whereas glycine betaine demonstrated a concentration independent effect. When the denaturant urea was used, a concentration dependent prolongation of the lag phase was observed, which indicated human IAPP stabilization in an aggregation incompetent state, and retardation of IAPP nuclei formation was observed. The effect was interestingly found to be fully compensated when the TMO was added in the molar ratio of 2:1 urea is to TMAO. However, a similar effect was not observed for glycine betaine, thereby indicating that direct interaction occurs between TMAO and urea. The amino acid proline was found to exhibit weak retardation of the elongation phase in a concentration dependent way, and also a reduction in IAPP fibril formation in a dose dependent manner. AFM measurements performed in the presence of proline revealed IAPP fibrils shortening and the formation of globular amorphous aggregates apart from the fibrillar assemblies. So these observations very well suggest that proline was able to divert the IAPP amyloidogenesis into a different aggregation pathway where formation of short and small fibrillar and non-fibrillar species occurs. A study also found that taurine in diet can diminish diabetes associated with dyslipidemia and hyperglycemia, but could not reverse the associated nephropathy. It is now clear that osmolytes would be of immense use in large number of human diseases. It is now important to look for strategies for the tissue-specific delivery of osmolytes. Since osmolytes are produced under different disease conditions, identifying specific osmolytes upregulated or downregulated under particular diseases will be useful for the selective use of osmolytes against a disease. This will benefit for the use of osmolytes as a strategy for diagnostic purposes. Large volume of work carried out to investigate the effect of individual osmolytes aggregation behavior or misfolding of proteins is still confined to case-by-case analysis. Understanding the effect of one osmolyte against the different properties of protein aggregates, misfolding or protein folding pathway will yield several additional insights for the therapeutic intervention of conformational diseases. Importantly, the use of osmolyte mixtures may help to increase the efficiency or reduce the toxicity against the use of high concentrations of one osmolyte. These are some of the references which I have gone through for preparing this talk. Thank you.